Synthetic chaperones for Huntington's therapy. Huntington's disease, or HD, is a devastating neurodegenerative condition that progressively impairs the ability to think, move, and perform daily activities. Affecting thousands of lives worldwide, it is the most common monogenic neuronal disorder since it is inherited autosomal dominantly, with a prevalence of 10.6 to 13.7 individuals per 100,000. Now, imagine a brilliant scientist who has dedicated their life to unraveling the mysteries of molecular biology. Someone who once contributed groundbreaking research that expanded humanity's understanding of life itself, but now finds it difficult to steady their hand to pipette a single drop. This is the cruel reality of Huntington's disease. It does not discriminate, and it can claim even the brightest of minds, reducing their once vital contributions to echoes of the past. Our research is dedicated to individuals like them, scientists, teachers, artists, innovators, and every person whose lives are shaped by HD. These are the minds that push humanity forward, the hands that build our future. By developing synthetic chaperones like HSBB5, we aim to combat Huntington's disease and safeguard not only the contributions of these brilliant individuals but also their dignity and humanity. Huntington's arises from a mutation in the HTT gene. This mutation leads to an abnormal repetition of CAG triplets, producing a protein with an excessively long polyglutamine structure. This misfolded protein forms aggregates that disrupt cellular function, causing widespread neuronal damage. These aggregates affect multiple areas of the brain, including the caudate nucleus and putamen. This effect causes tissue loss in this duo, and that loss is filled up by lateral ventricles. Over time, this leads to progressive loss of coordination, uncontrollable jerking movements known as chorea, and severe psychological symptoms such as depression and anxiety. Cognitive abilities deteriorate, leaving patients unable to manage everyday tasks. The result: patients experience motor dysfunction, cognitive decline, psychological disturbances, and, ultimately, severe disability. Alarmingly, there is currently no definitive cure for Huntington's disease. Existing treatments only manage the symptoms. Let me explain with an example. In cells, proteins need to fold properly for normal functioning. But sometimes these proteins get mixed up and don't fold correctly, which are called misfolded proteins. These misfolded proteins can be aggressive and lead to various neurodegenerative diseases. This is where HSBB5 comes in. HSBB5 works like a protein repairman. It finds the damaged proteins and fixes them, just like a broken toy repairman. This way, the misfolded proteins don't stick together and cause chaos, and HSBB5 organizes them, so the cell's work doesn't get interrupted. So, HSBB5 acts like a hero working to restore order in the cell. This is where our project comes in. We explored the use of synthetic chaperone proteins, specifically HSBB5, to tackle the issue of protein aggregation. Unlike traditional approaches, our method focuses on synthetic chaperones, offering a protein-specific solution with minimal off-target effects. HSBB5, also known as alpha-B crystalline, is an important factor in regulating cellular protein homeostasis. It performs its function via its oligomeric structure, which allows it to stabilize misfolded proteins by attaching to them. This stabilization step prevents misfolded proteins from aggregating and disrupting cellular functioning. HSBB5 also regulates protein-protein interactions, which helps to ensure appropriate protein folding and trafficking inside the cell. By executing these roles, HSBB5 contributes to the integrity of cellular proteostasis networks, limiting the emergence of diseases related to protein misfolding and aggregation, such as neurodegenerative disorders. We aim to reduce the harmful effects caused by misfolded proteins, providing a foundation for innovative treatments for this devastating disease. Let's begin by understanding the role of chaperone proteins. Chaperones, also known as stress proteins, are naturally occurring molecules in our cells. They are activated during cellular stress conditions such as high temperatures or changes in pH. Their job is to ensure that proteins fold correctly. When proteins misfold, chaperones either help them refold into their proper shape or direct them for degradation. The chaperone protein we focused on, HSBB5, is particularly promising. It has been shown to interact with misfolded proteins and prevent their aggregation. By targeting the secondary effects of Huntington's disease, namely protein aggregation, HSBB5 could potentially alleviate some of the disease's most debilitating symptoms. But how do we produce synthetic chaperones? This brings us to our experimental approach. Our experimental process began with a design of primers to amplify the HSBB5 and GFP genes. These primers were carefully designed manually, ensuring high specificity and accuracy for our target sequences. The forward primer was 22 bases long with a melting temperature of 62 degrees Celsius, while the reverse primer was also 22 bases long with a melting temperature of 60 degrees Celsius. The target amplification product length was set at 1,442 base pairs. These parameters ensured that we could obtain the desired genetic sequences with minimal errors or off-target amplification. Following primer design, we used polymerase chain reaction (PCR) to amplify the HSBB5 and GFP genes. PCR is a powerful technique that allows us to produce millions of copies of a specific DNA sequence in just a few hours. The reaction mixture included primers, DNTPs, a buffer, and a heat-stable enzyme called Taq polymerase. The PCR process consisted of an initial denaturation at 95 degrees Celsius for 2 minutes, followed by 25 to 30 cycles of denaturation, annealing and elongation, and a final elongation step to ensure complete synthesis of the target DNA. To confirm the success of our amplification, we used agarose gel electrophoresis. The PCR products were loaded onto a 2 to 3% agarose gel, stained with ethidium bromide, and visualized under UV light. Bands corresponding to the expected size of the amplified products confirmed the accuracy of our primer design and amplification process. Next, we cloned the PCR product into a vector for further expression. The vector we selected, PIS2, is commonly used for gene expression in yeast cells and offers features like the GAL1 promoter and URA3 gene. This vector was prepared by cutting both the PCR product and vector DNA with the EcoRI and BAMHI restriction enzymes. These enzymes created cohesive ends that facilitated efficient ligation of the DNA fragments. In the culture preparation, yeast extract peptone dextrose will be in the medium with a single colony. After that, the culture will be grown by shaking it at the right temperature. After shaking the culture, it will be placed in the incubator. The measurements will be done by the time it reaches the cell density stage. After measuring them, the culture will be transferred to a larger flask and they will be shaken. Later on, the new flask will be incubated to get the culture in the harvesting stage. When it reaches the harvesting stage, it will be centrifuged and the cell pellet will be washed with H2O. At the resuspension stage, water will be added to adjust the volume. After adjustment, it will be resuspended and centrifuged. After ligation, the recombinant vector was introduced into competent Saccharomyces cerevisiae cells using the lithium acetate transformation method. This approach involved creating a mixture containing the necessary components to enable the uptake of DNA by the yeast cells. The prepared mixture was carefully combined with the cells, ensuring proper conditions for transformation. A heat shock step was applied to facilitate DNA entry into the cells, followed by cooling and further processing to prepare the transformed cells for subsequent applications. Once transformed, the yeast cells were plated on selective media lacking uracil to ensure that only cells carrying the URA3 containing vector survived, as the URA3 gene enables growth in the absence of uracil. The selected colonies were then transferred to a medium containing galactose, which induced the expression of the GFP and HSBB5 genes under the control of the GAL1 promoter. After sufficient incubation, successful transformation was verified by observing the fluorescence of GFP under a microscope, confirming that both the HSBB5 and GFP genes were correctly expressed and the transformation was successful. With our synthetic HSBB5 protein successfully expressed in yeast, we moved on to purification. Using NINTA columns, we isolated the HSBB5 protein based on its affinity for nickel ions. The purification process involved several steps, including washing to remove nonspecific proteins and elution to recover the target protein. The purified protein will be concentrated. After that, the Kumasi Brilliant Blue G250 dye will be added and it will change the color. Thanks to the color change, the protein concentration will be analyzed by measuring it with a spectrophotometer. Later on, needed chemicals will be added. After that, standard protein solutions with concentrations of 0, 20, 40, 60, 80, and 100 mg per liter tubes will be prepared, and the protein solution with different concentrations will be added to each tube. Optical density will be read by the spectrophotometer. After finishing, the procedure calculations will be done. To confirm the integrity and purity of the HSBB5 protein, we performed SDS page and Western blot analyses. Purified proteins will be separated based on their molecular weights using 10 to 12% polyacrylamide gel. Protein samples will be denatured with a loading buffer and run on the gel. This process is necessary to confirm the presence of the proteins and ensure they are of the correct size. After SDS page, proteins will be transferred to a PVDF membrane and incubated with HSBB5 antibodies. The membrane will be visualized using the enhanced chemiluminescence (ECL) method, and protein expression will be confirmed. This analysis is conducted to definitively demonstrate the presence of HSBB5 protein and revealed bands corresponding to the expected molecular weight of HSBB5, indicating successful production and purification. Once the HSBB5 protein was purified, we proceeded to test its therapeutic potential using an in vitro model of Huntington's disease. The HT22 mouse cell line was chosen for this purpose, as it provides a reliable model for studying neuronal function and protein aggregation. To simulate the disease environment, these cells were transfected with a mutant Huntington (MHTT) gene. This gene, characterized by an abnormally long polyglutamine sequence, produces a misfolded protein that aggregates within the cells, mimicking the pathological conditions of Huntington's disease. We introduced the HSBB5 protein into the transfected HT22 cells at a concentration of 10 g/ml. The cells were incubated for 48 hours to allow the HSBB5 protein to interact with the mutant Huntington protein and potentially mitigate its aggregation. After the incubation period, we assessed the effects of HSBB5 using two key techniques: thioflavin T staining. This method allowed us to visualize protein aggregates within the cells. Thioflavin T is a fluorescent dye that binds specifically to aggregated beta sheet structures, such as those found in misfolded Huntington proteins. The reduction in fluorescence intensity in treated cells indicated a decrease in protein aggregation. Western blot analysis. This technique was used to quantify the levels of aggregated and soluble proteins in the cells. By comparing the intensity of bands corresponding to aggregated proteins in treated and untreated cells, we were able to evaluate the efficacy of HSBB5 in reducing aggregation. The results were promising. Cells treated with HSBB5 showed a significant reduction in protein aggregation compared to untreated cells. This reduction highlights the potential of HSBB5 to alleviate one of the key pathological features of Huntington's disease. In conclusion, our work demonstrates the potential of synthetic chaperones like HSBB5 in addressing Huntington's disease. By combining cutting-edge recombinant DNA technology with synthetic biology, we take one step closer to offering new hope to patients and their families. For the future aspects of our project, the observation of the positive effects of the HSBB5 protein in an in vitro setting for Huntington's disease may reveal the potential benefits of this protein in the pathogenesis of the disease. At this stage of the study, the effects of HSBB5 protein on intracellular and extracellular mechanisms, its contribution to reducing disease symptoms, and its capacity to manage cellular stress can be examined in detail. Based on these findings, the study can be tailored to achieve cell-specific delivery using various transport systems. Innovative approaches such as genetic delivery systems, viral or non-viral, or nanoparticles, can ensure the effective delivery of the protein to targeted cells. Optimization of these delivery systems is a critical step to enhance the bioavailability and therapeutic effects of the protein. In the next phase, these systems and therapeutic protocols should be tested on in vivo models. At this stage, both the biological efficacy and long-term safety of the protein are evaluated in animal models. Huntington's disease models that exhibit the symptomatic and pathological features of the disease can be used to thoroughly analyze the neuroprotective effects of the protein, its capacity to improve motor function, and potential side effects. Positive outcomes from these experiments can lay the groundwork for transitioning to human clinical trials. In the early phases of clinical trials, the pharmacokinetics, pharmacodynamics, safety profile, and tolerability of the protein are assessed. In later phases, the focus shifts to therapeutic efficacy and long-term outcomes. This process could support the potential of HSPB5 protein as an innovative and effective biological agent in the treatment of neurodegenerative diseases such as Huntington's disease. Thank you for watching. If you have any questions or thoughts, feel free to share them in the comments below.